Uh, Thank you all for staying so late. We have been delayed by hour or so, and you have been patient enough to be staying here and listening to the talks. And I request you all to please join us for the gala dinner tonight. This is going to happen here from 7:30 p.m. onwards. Thank you. And we'll go to our next session, that is the industry session. To start with, we have the first talk, the role of indigenous novel beta-lactamase inhibitor enmetazobactam in combination with cefepime. To go through it, we have Dr. Manish Paul as a speaker. Thank you. Over to Dr. Manish Paul.
Good evening. I think it's quite late and we are already, I think, quite tired and fatigued in listening to so many lectures. But I thought let us have a brief introduction. Dr. PNB is there and who's been part of, I consulted him earlier also. Okay, I'll speak on introducing this molecule to you all. Enmetazobactam, it is invented at Orchid Pharma, Chennai in the year 2006. And then it was out licensed at phase 1, stage 2 Electra Therapeutics. And after Electra Therapeutics conducted all the 3 phases of the clinical trials, it got approved all over the world except India. But then now India has also approved it, and we have it already soft launched just on Monday, 25th in Delhi. This molecule is approved by US FDA, European Medical Agency, and also UK MHRA. Now, I put this flag over there. The only reason being, this is a proud moment for India because this is the first ever Indian invention that the US FDA has ever approved. We have so many molecules, so many companies working in anti-infective drug discovery or many drug discovery programs, and never have a molecule got approved by FDA. So that's a proud moment for us. And here is a picture of when we launched it on 25th of this month, November. Orchid will be the manufacturer of this molecule and the marketed as brand name of Orblicef. Orblicef means orchid beta-lactamase inhibitor in combination with cefepime. And then we have partnered with Cipla to market it under 2 more brand names as Cipinmet and ESBL CoSip. Well, we were 2 inventors, UP Senthil Kumar, the chemistry counterpart, and me, who led the program for almost 18 years now. It is equivalent to 2 gestational periods in the number of years, probably, right? Some of the major characteristics of this molecule are it is, as long as you know the properties of cefepime, I think we know it all. It's restoring the activity of cefepime. It doesn't augment it. It brings it back to life. It is the ESBLs which kill the molecule, cefepime, and N-metazobactam is the one which saves it back to life. So it is also positioned as a carbapenem sparing molecule. Well, these are, I'll skip many of the slides because the journey is too long due to want of time. Look at this, this is tazobactam, essentially. The structurally both are alike, except that we have a methylation on the right side, which causes it to become N-metazobactam. It's methylation. The president of this society did mention you're adding a ponytail to the molecule, just like for what happened earlier to other molecules. So when you combine a cefepime, you'll take a look at that. In cefepime as well, we have a methylated group there, which forms it as a zwitterionic. When it is a zwitterion, the penetration into the gram-negative cell wall is much easier, and both of them reach the site of action quite well. And that's the reason why we believe it's going to be a good combination. And when you tested, it did show us an amazing activity. It acts on all these enzymes are listed over here, there, including the KPCs, the OXA48s, and TEM, the SHV, and it is stable to AMC. Why we say stable to AMC is, and metazobactam is does not induce the production of AMC here, while cefepime itself is stable to AMC. Well, much more data. These are all the published data to show, bring in the confidence that N-metazobactam inhibits various types of ESBLs, including KPCs and OXAs. That's what this whole data shows about. These are the IC50 values, which shows how the enzyme is actually binding. What exactly happens between tazobactam versus N-metazobactam? Why the difference? We have it in the market, cefepime, tazobactam at 2 gram with 2 gram. Unlike cefepime and metazobactam, it is 2 gram with 0.5 grams of N-metazobactam. The reason is the binding of the molecule into the enzyme is so tight and multi-socketed, so it doesn't get out easily. So the turnover number, what we call it as, you will see N-metazobactam for SHV, it is 5. Only you need 5 molecules of the drug. Unlike tazobactam, it requires 15 molecules. So similarly, it goes up to KPC2, it goes 100 versus 500. This is to demonstrate that how strong the binding happens with N-metazobactam. Some of the structure, how we identified the molecule, but this slide will tell you the MIC is how we decided in 2006 the MIC against all these various strains. Our molecule shows amazing in vitro activity. And then literature was published. Robert Bonomo's team did a lot of work, and more than 7,000 strains have been screened. And you will see the last column, which shows cefepime and metazobactam is almost equivalent to other meropenem vaporobactam or cephea avibactam, etc. in its activity. Now, we position it as meropenem sparing or carbapenem sparing molecule. The simple reason being, look at these two studies which have been published, wherein I pointed out in red to show, in fact, cefepime and metazobactam is much more better in its MIC values compared to meropenem alone. So this could be used in critical care, especially instead of reaching out to a meropenem, you can directly go towards using cefepime and metazobactam or Orblicef for your choice of drug. We did the testings in the year 2006, 2008, right? That's the period. And now when we tested in the month of August, the molecule is so good that even for the evolved strains of the bacteria, the resistance pattern hasn't changed. It is still all these more than 300 strains we tested, and it was active against all the strains of India. And I will show you in this next slide, you look at the meropenem MIC at less than 0.12 and compare the same with N-metazobactam, it is equal at MIC 90. So which is brings another confidence that yes, in India we can use it as carbapenem sparing molecule. Well, how did we invent the molecule? Of course, of all the things, it was the in vitro models which everybody will show it works. But when we did the in vivo model, that's when we showed the translation by developing a novel model and showed that it is highly active. So Orblicef, we in settings with an elevated prevalence of ESBL producing Enterobacteriaceae, we prove this molecule to be a valuable carbapenem sparing option. That's a major thing we are telling about. Now there is a confusion about zidibactam. Zidibactam is also an amazing molecule. It's also beta-lactamase inhibitor. And the difference is that is also an antibacterial agent by itself, and it has inherent activity in addition to being a beta-lactamase inhibitor. So ours is an exclusively beta-lactamase inhibitor without altering any of the PK/PD parameters. Well, this is out licensing phase 1. Now I pointed 2 points in red there to demonstrate why EMA, European Medical Agency, approved this molecule or this combination for HAP and VAP without having clinical trials is for the lung penetration studies. So both the cefepime and enmetazobactam have equal lung penetration abilities. That's the reason it was approved for that. As phase 2, it showed amazing efficacy in the smaller population, and it went for 19 different countries except India, and it proved to be superior to the tazobactam combination. This is where you can see how it differentiates itself from piperacillin tazobactam in its test of cure. The microbiological eradication of the organism is beautiful with cefepime and N-metazobactam compared to piperacillin tazobactam combination. So in conclusion, as I already alluded, it's a fixed dose combination of 2 grams of cefepime with half a gram of N-metazobactam. So cost of goods is lower as it becomes further, and approved for CUTI, HAP, VAP, and also bacteremia induced because of these conditions, and pyelonephritis associated CUTI. It is, if you look at any hypersensitivity, it is similar to whatever you look for any beta-lactam class of molecules. You must be aware of that one. Other than that, there's nothing to worry about it. And dosage, As I mentioned, we have got 2 grams, 2.5 grams is the packaging, which comes as Orblicef from Orchid Pharma. And next time you want to have any molecule in critical care, just go for Orblicef instead of reaching out for a carbapenem. Thank you. Here I am. And I see a lot of liquid already moving into the hall, so I think attention span starts distracting now. So thank you very much.
Thank you, Dr. Manish. Any question to Dr. Manish is ready to take. If no questions, thank you, Dr. Manish. We'll go to our next talk. Recent updates on balanced salt solution solute study by Dr. Ajinkya. Over to Dr. Ajinkya.
A very good evening to all of you. I'm Dr. Ajinkya Rodge. I'm with Medical Affairs with Fresenius Kabi. Uh, First of all, I would like to express my heartfelt gratitude to ISCCM Hyderabad to give us this opportunity to present the Solute study. The focus of this conversation is the Solute study, which was conducted by ICCM and has been recently published in the ICCM journal in the latest issue. So I won't be spending too much time on the basics since we are lacking time. And as Dr. PVN Gopal already mentioned, there's an more interesting fluids waiting for us. So all know for IV fluid therapies for resuscitation, maintenance, or replacements, we have multiple options like crystalloids, colloids, and blood products. Out of crystalloids, it's known that normal saline is the most commonly used one, but normal saline is not that normal because it is acidic in nature, it's potentially hypertonic. Other electrolytes apart from sodium and chloride are not present in it, and the supraphysiological chloride levels can lead to hyperchloremic metabolic acidosis, which has been observed in several RCTs meta-analysis, and so that's a well-established fact associated with normal saline infusions. I won't go into the details since you all know this, but as I said, studies have been saying that hyperchloremic metabolic acidosis is quite commonly or frequently associated with normal saline infusions, and it is also known to increase the incidence of acute kidney injury or AKI. And this metabolic acidosis and hyperchloremia may lead to several complications. So in light of this, there came the balanced salt solutions or the balanced crystalloids, which are very much near in terms of their electrolyte concentrations, very much physiological and similar to that of the plasma. And these are several examples for that. We have Ringer's lactate, Carbolite, Plasmalite, and other ones. These are the electrolyte compositions where you can observe the electrolyte concentrations, osmolarity, everything is quite near to that of the plasma or the normal concentrations of the electrolytes. Also, many guidelines and consensus statement have recommended use of balanced salt solutions. Like if we refer the GIFTA-sub guidelines for surgical patients, they say that balanced salt solutions recommended for the crystalloid resuscitation or replacement. Then Surviving Sepsis campaign, the latest update in 2021, also recommend crystalloids as first line fluid for resuscitation in adult with sepsis or septic shock. In that, crystalloids also balance crystalloid to be preferred over normal saline. Coming to another German guidelines, isotonic solution must not be used as a volume substitute in intensive care medicine, and balanced electrolyte solution containing acetate or malate instead of lactate may be used as a volume substitute for ICU patients, owing to the lesser hospital stay and lesser ICU stay. Similar findings from the S3 guidelines. So owing to the aforementioned side effects or risks and several guideline recommendations, you all must have observed that balanced crystalloids or BSS which have a composition closer to human plasma have increased and may be used as an alternative to normal saline therapy. The use of fluids in India has been very varied, as we are a vast country and a varied country. There has been no data to date regarding the practices in ICUs regarding the IV fluid therapy in India. So to study this or to fill this knowledge gap, ISCCM conducted a nationwide study, a multicentric study, which evaluated balanced crystalloids versus normal saline regarding their use in the ICU patients on several parameters. And this is the first Indian study capturing such kind of data regarding IV fluid administration in ICUs in our country. So this is the solute study, salt-based or balanced solution trends existing in Indian ICUs, a multicentric prospective observational cohort acronymed as solute study. Multiple authors, several of them are from Hyderabad, and it was published in the recent issue of Indian Journal of Critical Care Medicine. So as I said, it's a multicentric observational study where ICUs from all over India were involved. So type and volume of different IV fluids administered in ICU for the first 3 days of patient's admission in ICU were captured in these parameters. Then indications for renal replacement therapy, renal outcomes, acute kidney injury, and the ICU as well as hospital length of stay and mortality data was also captured. Patients were divided into 3 groups: the normal saline group, Ringer's lactate group, and the balanced salt solution group. Uh, all 192 ICUs from all over India enrolled, out of which data was collected or used from 144 ICUs. Out of these, majority of the hospitals were private, and almost 3/4 of the hospitals were private, and 1/4 from the government hospitals. Hospitals across the country contributed data for this study. So in terms of results or findings related to acute kidney injury, patients who received normal saline as compared to ringer lactate or balanced crystalloid on all the days, that is day 1 to 3, had a higher incidence of acute kidney injury. Coming to the intergroup comparisons, individual group comparisons, when the incidence of AKI was higher in normal saline group compared to Ringer's lactate, as well as when it was compared to balanced salt solution. And in comparison between Ringer lactate and balanced crystalloid group, the incidence was higher with the Ringer lactate group. So previously, randomized control trials and meta-analysis have said that there's no difference as such in the incidence of AKI between groups. But contrary to this, the solute study found patients who received normal saline during the study period had a higher incidence of AKI compared to both Ringer's lactate as well as balanced crystalloids. Coming to another parameters like urine output, although statistically significant difference was not observed, it was lower in normal saline group compared to the other groups. Serum creatinine was higher in the normal saline groups compared to the Ringer lactate as well as the balanced crystalloid groups. This was a parameter which was for the first time evaluated in this solute study. So daily fluid balance, we all know the fluid balance is associated with various, as earlier elaborated in the tribate. So on comparing mean fluid balance between groups, balanced salt solution had the least cumulative fluid balance compared to Ringer's lactate as well as normal saline. So none of the studies have analyzed the type of fluid that affect daily fluid balance and its effect on incidence of AKI. The previous study which was done, they only associated a relation between a positive or a high fluid balance and increased incidence of AKI, but the type of fluid associated was not evaluated in that. Solute study was the first one to do it. So in comparison to normal saline group had a higher fluid balance and hence higher acute kidney injury. The length of stay for ICU as well as for hospital was least with the balanced crystalloid group for ICU length of stay as well as with the hospital length of stay. Coming to mortality, higher mortality in the normal saline followed by the Ringer's lactate and balanced crystalloid group. Intergroup comparison, the normal saline had higher ICU mortality compared to the other groups. So, chloride-rich fluids, when given in large volumes, have been known to cause metabolic acidosis, leading to AKI and maybe higher mortality. Previous couple of studies have showed hyperchloremia associated with increased all-cause mortality. So, solute study also documented a higher mortality and AKI with the normal saline group. The strengths of the study I can highlight as this is the largest database from Indian ICU spread across the country. Findings were different from the previously reported studies that could be due to a local practice pattern. And this is the first study worldwide that looked at the fluid balance of different fluids and correlation with AKI. This could form the basis of much bigger and randomized trials to validate the findings. Limitations: certain renal outcome data was missing, which and if it was available, it could have contributed further to the results. Study period was precisely defined, that is the first 3 days of ICU admission, because of which maybe findings could have been diluted. There was no means of source data validation. So to highlight this solute study, which is the first of its kind study done by ICCM, solute study highlights the most preferred fluid given in ICU is still normal saline. Balanced crystalloids or balanced salt solution are associated with reduced incidence of AKI as compared to normal saline. ICU and hospital length of stay is lower with balanced crystalloids, and the study supports the use of a balanced salt solution over normal saline as resuscitation and maintenance fluids in ICU. With this, I end my session. Thank you for your patient listening, and thank you for staying back till the last session. Once again, I express my heartfelt gratitude to ISCCM Hyderabad to give us this opportunity. Thank you.
Dr. Manish and Dr. Ajinkya, thank you so much for the excellent talks, and thank all the audience who are patiently there in the till the end of the session on the first day. So we'll be closing the sessions for this first day, and much more eager fluids are waiting for you as all speakers were insisting upon. So our gala dinner starts from 7:30. We would request all of you to join with your families and friends also. We'll be having DJ. You can relax yourself as well as cocktails. Thank you. So, Dr. Manish and Dr. Ajinkya, can you please come over? Bhavani sir. So I would request Bhavani sir to felicitate Dr. Ajinkya, please. Mm.
Ramesh.
I would request someone from Dr. Manish's team to receive the memento. Thank you, everyone. So 7:30, bar will be open.